Hey everyone, and welcome. So, if you're gearing up for the CSIR NET in life sciences, you know you're facing something massive. It's a true deep dive into the code of life itself. And the biggest question is always, where do you even start? Well, in this explainer, we're going to hand you a map to navigate it all.
Right. You open up the official syllabus, and there it is. 13 core units, and each one is just jam-packed with hundreds of different topics. Looking at it all at once can honestly feel like you're standing at the bottom of a giant mountain, wondering how you're ever going to climb it. That sheer scale, that's the first big hurdle for everyone.
So the million-dollar question is this, right? How do you take that intimidating mountain of information and turn it into a map you can actually follow? A map that shows you how everything connects. That's exactly what we're going to do today. We're going to reorganize all 13 of those units into just a few core, easy-to-understand themes. Here's our game plan. First, a real quick look at what the exam is all about. Then, we'll dive into the syllabus using our 4 new themes: life's building blocks, the machinery of life, the grand tapestry, and to wrap it all up, biology in action. First things first, let's get on the same page. The CSIR UGC NET, or the National Eligibility Test, is basically the main gateway for getting a Junior Research Fellowship, or JRF, so you can pursue a PhD. It also determines if you're eligible for lectureship positions in Indian universities. Bottom line, it's the key that unlocks a career in academic research in India.
All right, let's start drawing our map with the very first theme, life's building blocks. This is all about the fundamentals, the chemical and structural foundation that all of biology is built on, from the tiniest atoms all the way up to the complex organelles inside a cell.
So this foundational theme actually pulls together the first 2 official units from the syllabus. Unit 1 is all about the biochemistry, the molecules themselves, like proteins, lipids, and nucleic acids. And then Unit 2 takes those very same molecules and looks at how they get assembled into the actual working parts of a cell. Now here's a key idea from Unit 1, bioenergetics. And listen, this isn't just about memorizing pathways from a textbook. It's about really understanding the universal currency of energy in every single living thing. It asks, how does a cell get energy? How does it store it? And how does it spend it to just stay alive? This is literally the engine room of biology. And from that engine room, we move into the factory itself, which is what Unit 2 is all about. It's understanding the cell's internal architecture, right? The nucleus is like the main office with all the blueprints. The mitochondria are the power plants, and the cytoskeleton, that's the internal scaffolding that gives the cell its shape and even lets it move around. Okay, so we've got the building blocks. Now let's see how they work. We're shifting from the static parts to the dynamic processes. This is our second theme, the machinery of life, and it explores all the intricate systems that let organisms grow, respond to their environment, and reproduce. Now, this is the biggest section on our map by far, covering 5 whole units from the syllabus. We start with the core processes happening inside a single cell, then we zoom out to how cells talk to each other, how a whole organism develops from just one cell, and finally, how entire systems work in both plants and animals. I love this. When you put Units 6 and 7 side by side, you see these incredible parallels and differences in how life solves the same problems. Plants use photosynthesis to get energy, animals use respiration. They both have hormones, they both have transport systems, but the actual mechanics behind them are just wonderfully unique. And the key insight here, taken straight from Unit 3, is that this entire theme, this whole machinery of life, really comes back to the central dogma of molecular biology. How is information stored in DNA, then transcribed into RNA, and finally translated into the proteins that do all the heavy lifting? If you can master this, you've pretty much mastered the core machinery of life. Okay, time to zoom out. We've seen how individual organisms work, but now we need to look at the bigger picture. Our third theme is the grand tapestry. This is where we explore how life gets so diverse, how traits get passed down through generations, and how all these different organisms interact with each other and their world over huge stretches of time. You can see how these 4 units just weave together so perfectly. We start with inheritance biology, basically the rules of genetics. Then we look at the incredible diversity of life that those genetics have created. After that comes ecology, which is the study of how all that diversity interacts, and we wrap it all up with evolution, the big story of how it all came to be in the first place. Looking at a timeline like this from Unit 11 really helps make these huge ideas feel real. I mean, we're talking about the origin of the very first cells, the major geological eras of the planet, the explosion of multicellular life, and the specific evolutionary steps that led right to us. It puts everything in this incredibly deep historical context. And this table here, from Unit 10 on ecological principles, is like learning the grammar of nature. See, life isn't just about individual survival. It's a huge web of interactions. Competition, symbiosis, predation, these are the relationships that actually define entire ecosystems, whether it's a rainforest or a tiny pond. All right, for our final theme, we're going to get practical. This is biology in action, or what I like to call the researcher's toolkit. How do we know all the stuff we've just been talking about? Well, this part covers the methods we use to study life and the ways we apply that knowledge to solve real-world problems. This theme neatly groups the last 2 units of the syllabus. Unit 13 is basically your how-to guide. It details all the lab techniques like molecular cloning, PCR, and microscopy. And then Unit 12 shows you the why, how we use those techniques in amazing fields like gene therapy, cleaning up the environment, and creating transgenic crops. Let's take a process like molecular cloning from Unit 13. It can sound super intimidating, right? But when you break it down into 4 simple, logical steps, you isolate the DNA, you insert it, you introduce it into a host, and then you select for it, you realize it's just a really clear, powerful tool that any researcher can use. And this is exactly why those methods matter so much. This isn't just theory for an exam. Unit 12 shows how this knowledge is being applied to improve our health through genomics, to boost our food supply with transgenic plants, and to heal our planet with bioremediation. This is biology making a real, tangible difference in the world. So, this final theme isn't really a separate topic. It's the thread that runs through everything else we've talked about. The methods in Unit 13 are how we study the molecules from Unit 1, the physiology from Unit 7, and the evolution from Unit 11. It's the how behind all of the what. So there you have it. When you look at it this way, the CSIR NET syllabus tells a complete and, well, a pretty logical story. It moves beautifully from the smallest molecules to entire ecosystems, and it connects deep theory to practical, world-changing applications. It's not just a random list of topics. It really is a map of modern biology. And when you see the syllabus like this, it stops being some scary obstacle, and it becomes a landscape, just waiting for you to explore. The mountain is still there, yeah, but now you have the map. So the only question left is which part of this incredible story of life are you going to master first?